Hey everyone, and welcome. Have you ever stared at a nutrition label on a bag of chips or a carton of juice and just wondered, how do they know that? How do they pinpoint the exact number of grams of protein or fat? Well, that's exactly what we're going to do today. We are pulling back the curtain and stepping right into the food science lab to find out.
So let's frame our whole journey around this one question. We start with something super familiar, right? A potato, straight from the earth. But our goal is to turn this thing into pure data, the numbers you see on that nutrition label. So, what's the secret? How does that incredible transformation actually happen?
You guessed it, it's not magic, it's science. And it all goes down inside a food analysis lab. So go ahead, put on your imaginary lab coat, and let's go inside to see how scientists take apart our food, literally molecule by molecule. Okay, before a single test gets run, before anyone even thinks about touching a food sample, the number one absolute top priority is safety. It's the most critical ingredient in any lab. And this isn't just about keeping the scientists safe, it's about making sure the results are pure and accurate. Now, here's the cool part about all this gear, the PP. It's got a two-way job. Sure, the lab coat and goggles are there to protect the scientists from, chemical splashes, but the gloves and the mask, they're just as important for protecting the food from the scientist. Even the tiniest bit of contamination could totally wreck the results.
And wow, this really shows you just how serious they are about it. This line is pulled directly from a real lab safety manual. It's not just about, oh, I don't feel good today. It's about the microscopic risk that germs from a simple cold could get into a sample and ruin a whole day's work. I mean, the level of care here is just intense.
All right, with our safety briefing done, we can get down to the main event, something called a proximate analysis. It sounds super technical, but it's basically the standard playbook for figuring out the major building blocks of any food. We're going to follow it step by step to completely deconstruct our potato.
First up, moisture. Now, this might sound kind of boring, but trust me, it's the foundation for everything else. A scientist will weigh a sample of potato with incredible precision, pop it into a special hot air oven cranked to 105 degrees Celsius, and just let it bake for hours. They weigh it again, and that difference in weight, that's all the water that's evaporated. And the result is pretty wild. Get ready for this. Almost 83% of a raw potato is just water. Yeah, water. And knowing this number is absolutely critical because every other calculation for protein, for fat, you name it, is based on what's left over, the dry stuff. Next up, we find the ash content. Now, that sounds weird, I know, but ash is just the sciencey term for minerals. To get it, the dried potato sample goes into something called a muffle furnace. This thing is like a pizza oven from the future, getting up to 600 degrees Celsius. It literally incinerates all the organic stuff, the carbs, the protein, and what's left behind? That's the ash. Those are your essential minerals like potassium. Okay, so how do you pull out just the fat? For this, they use this really elegant piece of lab equipment called a soxalate extractor. The best way to picture it is like a super-powered sci-fi coffee maker. But instead of hot water dripping through coffee grounds, you have a solvent that's constantly washing over the potato sample, dissolving and carrying away all the fat into a little flask at the bottom. Okay, buckle up, because measuring protein gets a little more intense. It's done with a classic three-part chemistry process called the Kildall method. First, they digest the sample with sulfuric acid. This converts all the nitrogen hiding in the protein into a different chemical form. Then they distill it, which turns that nitrogen into ammonia gas. And finally, they capture and measure that ammonia gas with insane precision. It's a whole process, but man, is it accurate. Now here's the kicker. They don't actually measure the protein directly. They measure the nitrogen, and then they do some math. And this is where it gets really interesting. The math is different depending on whether you're testing, say, a steak or a potato. Why? Because animal proteins are typically about 16% nitrogen, but plant proteins are a little denser, around 18%. So you have to use a different conversion factor to get the right protein number. And after all that heating and burning and distilling, what do we get? We get this. This is our potato, totally deconstructed. We directly measured the water, the ash, the fat, and the protein. And that big chunk left over, the carbs, that's actually figured out by subtraction. It's simply everything that isn't the other four things. And there you have it, the raw data for a nutrition label. But food is so much more than just those big 5 components on a label, right? I mean, what about flavor? What about that tangy zip or the sweetness? Food scientists measure that stuff too because it has a huge impact on quality and how long food stays fresh.
You ever see the word Brix on a juice carton and wonder what it is? It's basically a measure of sweetness. And how they measure it is just plain cool. They use a device called a refractometer. You put 1 drop of juice on it, and it shoots a beam of light through it. The more sugar in the juice, the more that light beam bends. It's a super clever and fast way to use the physics of light to figure out how sweet something is.
And then there's pH. You probably remember this one from school. It's the scale of acidity. And in food, this is a huge deal for 2 big reasons. Reason 1, taste. That sharp, tangy flavor in lemonade or yogurt, that's all thanks to acidity. But reason 2, and this is maybe even more important, is safety. High acidity creates an environment where many nasty little microbes just can't survive, which is one of the oldest and most effective ways we have to preserve food. So, okay, we can break down a potato in a lab. So what? Why does all this data matter? Well, this is where it gets really cool. Because once you understand the basic building blocks of food, you can start to influence how it's grown, and that takes us out of the lab and into the fields, into the amazing world of precision agriculture.
First up in the high-tech farmer's toolkit, sensors. We're talking real-time data streaming right from the field. Optical sensors can scan plant leaves from afar, and soil sensors can check moisture and nutrient levels constantly. They can spot a problem like a zinc or iron deficiency way before a human eye ever could. So once the sensors find a problem, how do you fix it efficiently? That's where drones come in. Think of them as little agricultural surgeons. They can fly over a huge field, use their cameras to pinpoint the exact weak spot, and then deliver a super targeted dose of the specific micronutrients needed right onto those plants. No waste, just pure efficiency. This goes even deeper, literally. By taking soil samples from all across a field and tagging them with GPS data, you can create a detailed nutrient treasure map. It shows exactly where the soil is rich and where it's lacking. This means farmers can add amendments only where they're needed, saving money and helping the environment. It's incredibly precise. And of course, the final piece of this whole puzzle is water. I mean, you can have all the nutrients in the world in the soil, but if the plant can't drink them up, it doesn't matter. Smart, controlled irrigation systems use sensors to make sure the soil moisture is always perfect, ensuring those nutrients are dissolved and ready for the plant's roots to absorb. So, look how far we've come. We went from a single potato to a full nutritional breakdown and all the way to a high-tech vision for the future of farming. We have learned to read the very language of our food with unbelievable precision. The only question left is, now that we can decode it, what story are we going to choose to write next?